Today we are going to discuss about the different types of Vacutainer tubes. So the Vacutainer tubes have a spray-coated silica and are used for the serum determination in chemistry. The samples processed in these tubes may also be used for the routine blood donor screening, immunohematology, and diagnostic testing of serum for infectious diseases, and also used for checking the blood glucose and bone marrow or coagulation studies. So the Vacutainers have a specific cape with different colors, and depending on the colors and the additives that are present inside the Vacutainer, we put the blood into different Vacutainers according to the studies we are going to process. So for example, we have a different, in front of us we can see, and the red, gray, purple, blue. So all these Vacutainers, they have different additives added into the Vacutainers and different color coding. So the first one that we can see here is the red cape Vacutainer, and it also called as plain Vacutainer because there is no anticoagulant present in this tube. This tube is completely empty, so there is no additive or there is no anticoagulant. So this plain Vacutainer is used for serological examinations in biochemistry, and it's also used for studies of the traces element present in the blood. Next one is the gray color, is also called as the sodium fluoride Vacutainer. So the additive present or the anticoagulant present in this Vacutainer is sodium fluoride, which prevent the blood from coagulation, and this Vacutainer is used for glucose estimation. The next is purple cape Vacutainer, is also called as Edta Vacutainer because the anticoagulant present in this Vacutainer is Edta, and the purpose of this Vacutainer in laboratory is it is used for hematological examinations like complete hemogram, ESR, and other studies. The light blue color Vacutainer contain 3.2% sodium citrate, and this sodium citrate precipitate or utilize the calcium present in the blood. So once it precipitate the calcium, there will be no activation of the coagulation pathway. So this Vacutainer is usually used for the studies of coagulation, or for the coagulation studies, like if we are going to measure PT for the extrinsic pathway, APTT for the intrinsic pathway. So for the coagulation studies, we usually use the light blue cape Vacutainer. The green cape Vacutainer contain anticoagulant heparin, and this is used for bone marrow studies. The next yellow cape or mild orange cape Vacutainer containing citrate, and this is also used for the blood culture because it also contains citrate that use the calcium. So here we can see not only the cape of Vacutainer, but we can also see the tube itself, and they have written that which type of anticoagulant or the additive present in these Vacutainers. So look at one by one. So this one is SST Vacutainer, SST tube Vacutainer, and here we can see that it is the sterile tube gel clot activator. Green cape written here heparin additive present. The black cape use or we can say fluoride Vacutainer is used for the glucose studies. The purple cape is Edta, it contains Edta. The red with no any additive or no anticoagulant, so it's a plain Vacutainer. Light blue containing sodium citrate anticoagulator, and the black cape is used for ESR. That's why it is also called as ESR tube. Now we are going to study about some of the Vacutainers in great detail. We will study the disadvantages, the advantages, and the uses of these Vacutainers. Number first one is that of the Lavender Edta tube, is also called as Edta or Lavender Edta tube. It contains Edta as an anticoagulant to prevent the clotting by chelating calcium that is present in our blood, so that is required for the coagulation process. So this Edta is chelate the calcium in the blood, so there will be no activation of our coagulation pathway. The disadvantage of this is excess amount of Edta can cause cell shrinkage and degeneration. Another disadvantage is that Edta induce exposure of platelets antigen, resulting in platelet clumping. Where we use the Edta tube, we can use for the CBC, ESR, malaria parasites, reticulocyte count, and parasite tests. The next one is the sodium citrate tube. The sodium citrate tube containing sodium citrate is an anticoagulant that prevent the blood clotting by precipitating calcium. So Edta was again doing the same. It's that for Edta tube, that was chelating the calcium, and this one, the sodium citrate tube, it is precipitating the calcium. So in both tubes we can say that they prevent the calcium from the activation of the coagulation pathway. So no calcium, no activation of the coagulation pathway. So it's important, so for the sodium citrate, it is important to fill the tube to the recommended volume for accurate coagulation studies result. That's why we can see that when the they nurses take blood sample, they used to fill the tube up to a given or recommended level, so it will be easy for them to do a good coagulation studies. The advantages of the sodium citrate tube is it prevents platelet clumping from platelet for the platelet count. So it prevents the platelet from clumping, and but EDTA tube we can see there will be clumping of the or aggregation of the platelets, while in case of sodium citrate the platelets count, there will be no clumping of the platelets, so there will be no change in the platelet count. And another advantage is that it allows coagulation studies is it removes the calcium ion from soluble calcium citrate form. So it helps in the formation of the calcium citrate complex, so it will allow us to study the coagulation. The disadvantage of this tube is it cannot be used for FBC or CBC because of the dilution factors, and it can be it can be used for APTT, PT, and DIC, D-dimer, fibrinogen, and platelet count studies. So the next one is green heparin Vacutainer, normally used for the clinical chemistry tests that requires plasma. Heparin is an acid mucopolysaccharide that inhibits the activation of prothrombin, or we can say the formation of thrombin from prothrombin, therefore stopping formation of fibrin from fibrinogen that prevent blood clotting. So if you know the pathway that our activation, extrinsic pathway as well as the intrinsic pathway that causes activation of prothrombin to thrombin, and this thrombin then activate fibrinogen to fibrin. So factor 2 converts to factor 1. Then there will be a secondary thrombi formation, secondary hemostasis, that further provides stability to the thrombus. So heparin, it inhibits this pathway, so there will be no activation of thrombin from prothrombin, so there will be no formation of clot. So it inhibits the formation of blood clot. The disadvantages are it cannot be used for the hematological tests as it causes cell clumping and gives a blue background to the blood film. The uses of this heparinized tube, as we said, it's used for bone marrow tests and coagulation. It helps in beta hydroxybutyrate studies. Next one is gray tube or the fluoride tube. It containing sodium fluoride is an enzyme that inhibits continued metabolism of glucose, hence preserve blood glucose concentration. So it is a glucose tube, we can say that. This gray head tube or the fluoride Vacutainer, so that's why it is used to steady blood glucose concentration and after we taking the blood, so it's used for the glucose estimation. We can say that for the fasting glucose or postprandial glucose. So the next tube is red cape Vacutainer or the plain Vacutainer. So we know that this plain Vacutainer is used for the serological examinations. So for the serological examinations we only need the serum of the patient, but if we cannot take only serum, so once we take blood we inject the blood into this Vacutainer, and hence this Vacutainer does not contain any anticoagulant, so it helps in the rapid coagulation process. So there will be coagulation and of the cells, the blood cells, so it easily separate from the plasma, and after that we can take the plasma for the serological studies. So it speed up the clotting process and aid separation of blood serum from the blood cells, so it will be easy for us to study the serological examinations. Thank you.